This is a homozygous bull, white in color, with a genotype W, and this is a homozygous cow, red in color, with a genotype R. What color will their calf be in the F1 generation? Well, it isn't white or red, and neither is it a color in between. The calf is roan colored. Its coat is a mix of red and white hair. Therefore, in this case, the F1 generation doesn't resemble either of the parents as in dominance, nor is it in between as in incomplete dominance. What the calf displayed was codominance. Codominance refers to a situation where a heterozygous organism has a phenotype that demonstrates traits from both dominant as well as recessive genes. A codominant trait is not blended but is independently and equally expressed. Another widely known example of codominance in human beings is the ABO blood system, which is used to determine human blood type. The plasma membrane of the red blood cells, or the RBCs, present in human blood, has sugar polymers that protrude from its surface. It is these sugar polymers that determine the specificity of the major blood types. For example, the difference between blood types A and B lie in a single sugar unit that protrudes from the end of a carbohydrate chain of a glycoprotein or glycolipid on the plasma membrane of an RBC. It is the gene I that is responsible for determining the type of sugar produced by the RBCs. This gene has three different alleles, namely IA, IB, and small I. Of these, alleles IA and IB produce sugars that are slightly different from each other, while the allele small I doesn't produce any sugar. Here, the alleles IA and IB are completely dominant over allele small I. Now, human beings are diploid organisms and hence possess only two of the three alleles or the gene I. Consequently, as you can see, there are 6 possible combinations of alleles. Therefore, if the allele from one of the parents is IA and from the other is small I, only IA is expressed in the offspring, as small I does not produce any sugar. In this case, the RBCs have only type A sugar in them, and the genotype of the offspring is IA small I, and the phenotype or the blood group is A. Similarly, when the allele from one of the parents is IB and from the other is small I, only IB is expressed in the offspring, as the RBCs have only type B sugar in them. In this case, the genotype is IB small I, and the phenotype is B. However, when the allele from one parent is IA and from the other is IB, both the alleles express their own types of sugar in the offspring due to codominance. In this case, the RBCs have both A and B types of sugars in them, and the genotype of the offspring is IA, IB, and the phenotype or the blood group is AB. Now, if the alleles from both parents are IA Once again, the RBCs have only type A sugar in them, and therefore, the offspring has a genotype IA, and phenotype A. Likewise, if the alleles from both parents are IB, the RBCs have only type B sugar, and so the offspring has a genotype IB, and phenotype B. However, if the alleles from both parents are small I, the RBCs have no sugar in them, and so the genotype of the offspring is small I, and the phenotype is O. Therefore, as there are 3 different types of alleles, there are 6 possible genotypes and 4 different blood groups or phenotypes that exist. Apart from being a good example for codominance, the ABO blood grouping system is also an ideal example of multiple alleles. As we just saw, the gene I has 3 alleles, IA, IB, and small I, that govern the same character, that is, blood group. However, since an individual can have only 2 alleles, it is possible to learn about multiple alleles only when detailed population studies are conducted. According to the multiple alleles concept, more than 2 alleles govern the same character. Now, let us study an experiment that shows how a single gene may sometimes produce more than one effect. Pea seeds have a single gene with 2 alleles, B and small b, that control starch synthesis in them. In the experiment conducted, effective starch synthesis was seen in B homozygotes, and the seeds produced were large or had a large amount of starch in them. Moreover, the pea seeds were round. On the other hand, small b homozygotes were not very efficient at synthesizing starch, and so the pea seeds produced were small or had less starch in them. Also, the seeds were wrinkled. Moreover, in the experiment conducted, the seeds or the starch grains produced by heterozygotes B were of intermediate size, but were round. Now, in this experiment, if we consider the starch size as the phenotype, then the alleles are displaying incomplete dominance. On the other hand, if we consider the shape of the seed as the phenotype, then the alleles are displaying dominance, as the seeds were round like one of the parents. Hence, the experiment shows that dominance is not an autonomous feature of a gene or gene product. Therefore, dominance, incomplete dominance, or codominance depends on gene product and the production of a particular phenotype from this product. It also depends on the phenotype that we wish to examine in case the same gene influences more than one phenotype.